Hi students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is chromosome and cell cycle. Cell cycle we'll be studying in the next section of my presentation. Here we will be studying about the chromosomes. Okay. So what is a chromosome? Chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Each chromosome is made up of protein and a single molecule of deoxyribonucleic acid, that is DNA. Okay, they are passed from parents to offsprings. DNA contains the specific instructions that make each type of living creatures unique. Okay, the term chromosome comes from a Greek word for color, means chroma. Okay, and body means soma, chromosome. Okay, this is chromosome. Chroma means colored and some means body. Okay, they gave this term or name to the chromosomes because they are cell structures or bodies that are strongly stained by some colorful dyes used in research. Okay, now the question arises, what do chromosomes do? Okay, the unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called histones. Without such packing, DNA molecules would be too long to fit inside the cell. For example, if all of a DNA molecules in a single human cell were unwound from their histones and placed end to end, they would stretch 6 feet. I repeat, 6 feet. If all of the DNA molecules in a single human cells were unwound from their histones and placed end to end, they would stretch 6 feet. Okay, for an organism to grow and function properly, cells must constantly divide to produce new cells to replace old worn out cells. During cell division, it is very essential that DNA remains intact and evenly distributed among cells. Chromosomes are a key part of the process that ensures DNA is accurately copied and distributed in the vast majority of cell divisions. Still, mistakes do occur on rare occasions. Changes in the number or the structure of chromosome in new cells may lead to serious problems. For example, in humans, one type of leukemia and some other cancers are caused by defective chromosomes, which are made up of joined pieces of broken chromosomes. It is also crucial that reproductive cells such as egg, sperm, they contain the right number of chromosomes and that those chromosomes have the correct structure. If not, the resulting offsprings may fail to develop properly. For example, people with Down syndrome and Turner syndrome, such as Down syndrome, they have 3 copies of chromosome number 21 instead of the 2 copies found in the other people. Okay, so do you know, students, what is so special about chromosomes? They are huge. That means one base pair is equals to 600 Daltons. An average chromosome is 107 base pair long and is equals to 1 to 10 Daltons. For comparison, a protein of 3 into 10 to 10 phi is considered very big. They contain a huge amount of non-redundant. Non-redundant means repetition. Okay, so they contain a huge amount of non-redundant information. It is not just a big repetitive polymer, but it has a unique sequence. Okay, I have already introduced chromosomes to all of you. So once again, chromosomes are the structure that contain the genetic material. They are complexes of DNA and protein. The genome comprises all the genetic material that an organism possesses. In bacteria, it is typically a single circular chromosome. In eukaryotes, it refers to one complete set of nuclear chromosomes. Okay, eukaryotes, they possess a mitochondrial genome, and the plants, they also have a chloroplast genome. Okay, in continuation to that, the main functions of the genetic material is to store information required to produce an organism. That is, the DNA molecule does that through its base sequences. Base sequences are necessary for synthesis of RNA and cellular proteins. Second, they help in the proper segregation of the chromosomes. They help in replication of chromosome, and they help in compaction of the chromosome. Compaction of the chromosome we will study in the later presents. Okay. So, what do you think? Do all living things have the same type of chromosome? No. Chromosome, they vary in number and the shape among living things. Okay. Most bacteria have 1 or 2 circular chromosomes. Human, along with other animals and plants, they have linear chromosomes that are arranged in pairs within the nucleus of the cell. The only human cell that do not contain pairs of chromosomes are reproductive cells or gametes, which carry just 1 copy of each chromosome. When 2 reproductive cells unite, they become a single cell that contains 2 copies of each chromosome. This cell then divides, and its successors divide numerous times, eventually producing a mature individual with a full set of paired chromosomes in virtually all of its cells. Okay? Beside the linear chromosome found in the nucleus, the cell of the humans and the other complex organisms, they carry a much smaller type of chromosome similar to those seen in bacteria. This circular chromosome is found in mitochondria, which are structures located outside the nucleus that serves as the cell's powerhouse. Okay? Now we will study about the chromosome structure. During nuclear division, the DNA in a eukaryotic cell's nucleus is coiled into very tight compact structure called chromosome. Okay, these are rod-shaped structures that are made up of protein and DNA that I have already told you earlier, which are visible only during nuclear division. The DNA in eukaryotic cell is coiled tightly around proteins called histones, which help in the tight packing of DNA. During interphase, the DNA is not tightly coiled into chromosome, but they exist as a chromatin. Okay, and when preparing for the nuclear division, during the S phase of the interphase, the chromosome copy themselves. That is, the DNA replication occurs. Each half of the chromosome is now called as chromatid. Note that there is still 1 chromosome. It consists of 2 chromatid but has only 1 centromere. The function of this is to hold the 2 chromatids together until they separate during anaphase. Okay, chromosomes are simpler in prokaryotes. Their DNA is in a single chromosome and they exist as a loop. Okay. In eukaryotic cells, chromosomes are composed of single molecule of DNA with many copies of 5 types of histones, which are proteins. I have shown a chart of types of histone protein in the next slide. Okay, histones are the protein molecules that are rich in lysine and arginine residues that are positively charged. Hence, they bind tightly to the negatively charged phosphates in the DNA sequence. A small number of non-histone proteins are also present. These are mostly transcription factors. Transcription factors regulate which part of DNA to be transcribed into RNA by the process of transcription. Transcription you will be studying in the 12th standard zoology. Okay. During the S phase of the mitotic cell, the chromosomes are duplicated. At the beginning of the mitosis, the chromosomes are duplicated and they begin to condense into short structures which can be stained and observed easily under the light microscope. Okay. And these duplicated condensed chromosome, they are known as diodes. The duplicated chromosome, they are held together at the region of centromere. Centromere is a constricted region of the linear chromosome. And although this constriction is called as centromere, it is not located exactly in the center of the chromosome, and in some cases it is located almost at the chromosome end. Okay. The region on either side of the centromere are referred to as chromosome arms. Centromere, they help to keep chromosome properly aligned during the complex process of cell division. As chromosomes are copied in preparation for production of a new cell, the centromere, they serves as an attachment site for the two halves of the replicated chromosome, known as sister chromatids. Okay. This is centromere, this is P arm, this is Q arm, and this is telomere. They both are the telomeres. Okay. Each chromosome has a constriction point called as centromere. As I have told you, the centromeres in humans, they are made up of about 1 to 10 million base pairs of DNA, which divides the chromosome into two sections or two arms. Okay, the short arm of the chromosome is labeled as P arm, and the longest one is labeled as Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of the specific gene. Okay, now what are telomeres? Okay, what are telomeres? Telomeres are the repetitive stretches of DNA located at the end of the linear chromosomes. They protect the ends of chromosome in a manner similar to the way the tips of the shoelaces keep them from unveiling. Okay, in many types of cells, telomeres, they lose a bit of their DNA every time a cell divides. Eventually, when all of the telomere DNA is gone, the cell cannot replicate and thus it dies. Okay, white blood cells, that is WBCs, and other cell types with the capacity to divide very frequently have a special enzyme that prevents their chromosomes from losing their telomeres. Because they retain their telomeres, such cells generally live longer than other cells. Telomeres also play a role in cancer. About the cancer we will be studying in detail in the just after two presentations. Okay, the chromosomes of malignant cells usually do not lose their telomeres. It helps to fuel the uncontrolled growth that makes the cancer so devastating. Okay, how many chromosomes do humans have? Humans have 23 pairs of chromosomes for a total of 46 chromosomes. In fact, each species of plants and animals has a set number of chromosomes. Okay, a fruit fly, for example, has 4 pairs of chromosomes, while a rice plant has 12 and a dog has 39. Okay. Now the question arises, how are chromosomes inherited? In humans and most other complex organisms, one copy of each chromosome is inherited from a female parent and the other from the male parent. This explains why children inherit some of their traits from their mother and others from their father. Okay, the pattern of inheritance is different from the small circular chromosome found in mitochondria. Only egg cells and not sperm cells keep their mitochondria during fertilization. So mitochondrial DNA is always inherited during fertilization. So mitochondria DNA is always inherited from the female parent. In humans, a few conditions including some forms of hearing impairment and diabetes have been associated with DNA found in the mitochondria. Now come to the chromosome number. Each human somatic cell, okay, we are not talking about the genetic cell, we are talking about the somatic cell, means body cell. They contains 2 complete sets of chromosome. This is the normal for eukaryotes and is known as diploid. For humans, this means our cells have 46 chromosomes. For other species, this number can be very different. Okay, such as 94 in goldfish and 14 in lettuce. Sex cells have only one set of chromosomes, and such cells are known as haploid. Each cell is unique as it is produced by meiosis, though the mother cells for the gamete are produced by mitosis. The final division is always meiotic. Okay, that we will be studying in detail. Okay, Meiosis is necessary because most organisms, they reproduce sexually, and during fertilization the gametes fuse. That means sperm and the eggs, they fuse, and the number of the chromosome therefore doubles, okay, to produce the first cell of the next generation, that is 2n, okay. N plus n is equals to 2n. Chromosomes can be divided into two types. In humans, we have 22 pairs of autosomes, which are the same in every individual, and the one pair of sex chromosome. In females, it is found XX, and in males, it is XY. This means that females are the homozygous sex and the males are the heterozygous sex. The effect of this is that females can be carrier for sex-linked characteristics. I will explain this when we will study genetics, okay? The two matching sets of autosomes are known as homologous pairs, and both will code for the same feature, okay, but they may well have different alleles. Viral genome. Viruses are the small infectious particles that contain nucleic acid, that is DNA or RNA. Usually they contain RNA, nucleic acid, which is surrounded by a capsid of protein. For replication, viruses, they rely on their host cell, that is the cells they infect, okay. Most viruses, they exhibit a limited host range. They typically infect only specific type of cells of one host only, okay, one host species. A viral genome is a term used in a whole of the genetic material. The genome can be DNA, RNA, single-stranded or double-stranded, circular or linear. Viral genome vary in size from a few thousand to more than 100,000 nucleotides, okay. Now come to the general structure of viruses. This is non-enveloped virus. That means it does not have capsid, okay. It is a enveloped virus with spikes, okay. Bacteriophage, the bacteria that attacks viruses, is very common virus. It is a virus that attacks a bacteria, is known as bacteriophage. It is very common virus that consists of Na plus protein, which is usually enclosed by three types of capsid structure: icosahedral, filamentous, head and tail, okay. And eukaryotic virus, they consist of capsid plus Na, and capsid can be of isohedral and filamentous, okay. Now come to the bacterial genome. Bacterial chromosome DNA is usually a circular molecule that is a few million nucleotides in length. Escherichia coli, it has 4.6 million base pairs, and Haemophilus influenza has 1.8 million base pairs. A typical bacterial chromosome, they contain a few thousand different genes. Structural gene sequences account for the majority of the bacterial DNA, okay. This is the representation. The key features are most but not all bacterial species, they contain circular chromosomal DNA. A typical chromosome is a few million base pairs in length, as I have told you. Most bacterial species, they contain a single type of chromosome, but it may be present in multiple copies. Several thousand different genes are interspersed throughout the chromosome, and one origin of replication, ori, O-R-I, ori. This is origin of ori. Replication is required to initiate DNA replication. These are the repetitive sequences, short interspersed throughout the chromosome, okay. This is the circular chromosomal DNA that is found in bacteria, and this is the formation of the loop domains. The loop structure compacts the chromosome about 10 folds, okay. As you can see that it has formed folds, okay, with associated proteins. These are the proteins, okay, and these are the chromosomes. To fit within the bacterial cell, the chromosomal DNA must be compacted about 1,000 folds. This involves the formation of the loop domains, as I have told you. The number of the loops, they varies according to the size of the bacteria chromosome and the species. E. coli has 50 to 100 with 40,000 to 80,000 base pairs of DNA in each. Okay. When DNA helix has the normal number of base pair per helical turn, it is in the relaxed state. Now we are discussing about the supercoiling. Okay, when the DNA helix has the normal number of base pairs per helical turn, then it is in the relaxed state. Okay. Changing this normal amount of twist can be demonstrated by grasping both ends of the short linear module and twisting the ends in the opposite direction. If the helix is over twisted so that it becomes tighter, okay, and the edges of the narrow groove move closer together. If the helix is under twisted, the edges of the narrow groove move together. Okay, the helix is under twisted, the edges of the narrow groove move further apart. Okay, this is the representation how looped chromosomal DNA got converted into a supercoiled looped DNA. Okay, just remember that twisting the twist from a relaxed state, they requires adding energy and increases the stress along the molecule. Okay, as I have told you that over twisting, they leads to positive supercoiling, while under twisting, they leads to negative supercoiling. This is a chromosome. Okay, this is a chromatid. Okay, and this is a over twisting that has led to a positively supercoiled structure, and this is a relaxed state, and this is the negatively supercoiled structure because twisting been done. Okay. That means if the helix is under twisted, I'm talking about the negatively coiling. If the helix is under twisted, the edges of the narrow groove move further apart. Okay, and this leads to negative supercoiling. Now come to eukaryotic chromosome. Eukaryotic species, they contain one or more sets of chromosomes. Each set is composed of several different linear chromosomes. The total number of DNA in eukaryotic species is typically greater than that in bacterial cells. Chromosomes in eukaryotes, they are located in nucleus. To fit in there, they must be highly compacted. Okay, that chromosome compaction is my next presentation, so stay tuned for that. Eukaryotic genomes is vary substantially in sizes. In many cases, this variation is not related to complexity of their species. For example, there is a two-fold difference in the size of the genome in two closely related salamander species. Okay, the difference in the size of the genome is not because of the extra gene, rather the accumulation of the repetitive DNA sequences. Organization of eukaryotic chromosome. A eukaryotic chromosome, they contain a long linear DNA molecule. Three types of DNA sequences are required for chromosomal replication and segregation, such as origin of replication, that is ori, centromere, and the telomeres, the three units of a chromosome. As you can see, this is a telomere, this is the origin of replication, these both is the ori, okay, origin of replication. This is a centromere, and the protein which is associated with the centromere is a kinetochore protein. This is again ori, ORI, that is origin of replication, and this is again a telomere. And this is the P arm. Short arm is labeled as P arm and the longer arm is labeled as Q arm. Okay. This represents repetitive sequences and this represents genes. Okay. What are the key features? Eukaryotic chromosomes, they are usually linear. A typical chromosome is tens of millions to hundreds of millions of base pairs in length. Eukaryotic chromosome, they occur in sets. Many species are diploid, which means that somatic cells, they contain two sets of chromosome. Genes are interspersed throughout the chromosome. A typical chromosome contains contains between a few hundred and several thousand different genes. Each chromosome, they contain many origin of replications that are interspersed about every 1 lakh base pairs. Each chromosome, they contains a centromere that forms a recognition site for the kinetochore proteins, and these are the telomeres. They contain a specialized sequence located at both the ends of the linear chromosome. Repetitive sequences, these are the repetitive sequences. They are commonly found near centromeric and the telomeric region, as you can see. Okay, this is the centromeric region, and it is most evidently found near the centromere region, but they may also be interspersed throughout the chromosome. Okay, eukaryotic chromosome is packed in hierarchical levels mediated by various proteins. DNA duplex gets converted into nucleosome, and nucleosome then converts into a supercoiled chromatin, and the chromatin gets converted into chromosome finally. Okay, as you can say that this is a DNA duplex, so short region of the DNA double helix, which is about 2 nanometer long. Okay, and it's like a beads on the string, motiyon ki mala. Okay, they form a chromatin. It's about 11 nanometer long. Okay, and this is 30 nanometer chromatin fiber of the packed nucleosome, which is about 30 nanometer. Okay, and this forms solenoids. Okay, and sections of the chromosome in an extended form, which is about 300 nanometer. As you can see that supercoiling is going on and on and on, and thus it forms entire mitotic chromosomes, which is about 1400 nanometer in length. Okay, so this comes to an end. In the next presentation we will be studying about the chromosome compaction. Okay, till then stay tuned and keep watching Edupedia Word videos. Thank you.